Can we rewrite the very fabric of life? Could we cure genetic diseases before birth? Welcome to the future of medicine. Imagine a world where we can edit genes as easily as typing on a keyboard, where we can eradicate hereditary diseases from the human gene pool, where we augment our bodies to become more resilient, more adaptable. This is not science fiction. It's the promising reality of genetic engineering, and at the forefront of this revolution is a tool called CRISPR. CRISPR, or Clustered Regularly Interspaced Short Palindromic Repeats, is a groundbreaking technology that allows scientists to edit genes with unprecedented precision. By harnessing the power of this tool, we could potentially transform medicine and healthcare as we know it, paving the way for cures to diseases that have plagued humanity for centuries. So, are you ready to explore this brave new world? Let's delve into the fascinating world of CRISPR gene editing. A CRISPR, or Clustered Regularly Interspaced Short Palindromic Repeats, is more than just a mouthful. It's a game changer for genetic engineering. Imagine a pair of microscopic scissors that can precisely cut and paste pieces of DNA within an organism's genome. That's essentially what CRISPR technology allows scientists to do. CRISPR was first discovered in bacteria as a defense mechanism against viruses. The bacteria would capture snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. These arrays help the bacteria recognize and defend against the virus if it attacked again. Fast forward to the 21st century, and scientists have harnessed this bacterial defense mechanism to edit genes in any organism, including humans. The process begins with a molecule known as guide RNA. This molecule is designed to match the sequence of a specific gene that needs to be edited. Think of the guide RNA as a GPS system, leading the way to the target destination. In this case, the target is the gene that needs to be edited. The guide RNA binds to this gene and signals for the next player to step in. The Cas9 protein. Cas9, known as a molecular scissors, is an enzyme that can cut DNA strands. Once the guide RNA has located and bound to the target gene, Cas9 comes in and makes a precise cut in the DNA. After the DNA is cut, the cell's natural repair machinery kicks in to repair the break. Here's where the magic happens. Scientists can provide a template for how they want the DNA sequence to look after repair. This can involve adding, deleting, or replacing certain DNA sequences. And voila! The gene has been edited. The potential applications of this technology are staggering, from curing genetic diseases to improving crop yields and beyond. However, it's important to remember that gene editing is a powerful tool and should be used responsibly. It's like the old saying goes, with great power comes great responsibility. As we continue to explore the possibilities of CRISPR, we must also consider the ethical implications and strive to use this tool for the betterment of all living beings. CRISPR is a powerful tool, but with great power comes great responsibility. From curing genetic diseases to improving agricultural practices, CRISPR holds immense potential. Let's begin with medical treatments, where CRISPR is making waves. In the realm of genetic diseases, CRISPR is seen as a beacon of hope. Imagine being able to snip out the genes causing cystic fibrosis, muscular dystrophy, or even Huntington's disease. The beauty of CRISPR is that it makes this possible. It's like having a molecular scalpel that can precisely target and remove flawed genes, potentially eradicating these diseases from our genetic blueprint. But it's not just about eliminating diseases. CRISPR could also enhance our quality of life. We could potentially slow down aging, improve our physical abilities, or even boost our resistance to viruses. Picture a world where we could customize our own genetic makeup, like choosing traits in a character creation screen of a video game. That's the kind of power CRISPR could give us. Moving beyond the human body, let's look at how CRISPR is revolutionizing agriculture. We've been selectively breeding plants and animals for centuries to get desired traits, but this is a slow and unpredictable process. CRISPR speeds things up, allowing us to directly edit the genes of crops to make them more nutritious, resistant to pests, and able to withstand harsh climates. This could be a game changer in addressing food security issues as our global population continues to grow. Looking ahead, CRISPR could even help us combat climate change. Scientists are exploring the idea of using CRISPR to modify the genes of certain bacteria, enabling them to capture more carbon dioxide from the atmosphere. But let's not get carried away. While the potential of CRISPR is staggering, We're still in the early days of understanding and implementing this technology. There are many legal, ethical, and safety considerations to navigate, and like any powerful tool, CRISPR could be misused if it falls into the wrong hands. The possibilities with CRISPR seem endless, but it's not all sunshine and roses. With the power to edit genes, we tread on ethically complex grounds. Should we play God? This isn't a question we can dismiss lightly. CRISPR, as innovative and promising as it is, opens up a whole new Pandora's box of ethical considerations. Let's take a moment to delve into this. On one hand, the potential of CRISPR is immense. It could eradicate genetic diseases, improve crop yields, even help us fight climate change. But with great power comes great responsibility. The misuse of CRISPR technology is a genuine concern. Imagine the consequences if it falls into the wrong hands. The creation of harmful biological agents, the potential for a new kind of biological warfare is a chilling prospect. Then there's the question of designer babies. If we have the ability to edit our children's genes, where do we draw the line? Should we only use it to prevent disease, or is it ethical to enhance traits like intelligence or physical abilities? This is uncharted territory with no easy answers. And let's not forget the potential for unintended consequences. We're still learning about our own genome. What if an edit meant to fix a problem ends up causing a new one? The ripple effects could be far-reaching and unpredictable. So, as we embrace the possibilities of CRISPR, we must also acknowledge the ethical challenges it presents. We need to have these conversations to establish guidelines and safeguards. As we stand on the brink of a new era of medicine, we must tread carefully. The CRISPR revolution is here, promising a future of unimaginable possibilities and complex ethical dilemmas. We've explored the science behind it, its applications from curing diseases to improving our food supply, and the ethical considerations it brings. The potential to transform medicine and our world is immense. However, with great power comes great responsibility. We must tread this new path with caution and wisdom. The CRISPR revolution is upon us, and it's up to us to steer it towards a better tomorrow.